Hi, I'm Mick Clews. Welcome to AUREUS. I come to AUREUS from Australia. I'm a fourth generation dairy farmer, was in general practice, in dairy practice in Australia before I came over here 6 seasons ago. So one of the things I learnt when I came to New Zealand from Australia and back into practice from dairy farming was the little things that were different about New Zealand dairy farming compared to Australian dairy farming. So one of the things that I had to get used to was the concept of spring mastitis being caused by strep uberis and summer mastitis being caused by Staph aureus. One of the things that we wanted to find out for ourselves, or particularly I wanted to find out, was if that was the case, that strep uberis mastitis was the predominant mastitis caused at calving or in that first peri-calving period, and Staph aureus came in later, when was that cutoff? Was it after a week? And every different person you spoke to told me a slightly different version. Some people would say Staph aureus mastitis started to predominate after only a month or after only a week, or some people would say that it changed at Christmas time. So what we wanted to do with the study was to find out when were the different bugs causing mastitis prevalence in our particular area. We wanted to repeat that. If we knew that strep uberis is very sensitive to just about any antibiotic, that we should use the most responsible choice for strep uberis antibiotics, and then only change to something stronger and longer when we were required to. So we had to work out when that period was, firstly, and secondly, get some reassurance that that protocol was acceptable to the farmers, and that the results that we got out of that protocol, whilst we knew they were going to be more responsible, actually came up with a similar or better clinical cure rate to what we would get through the old system. So we asked farmers to bring in a milk sample from every case of mastitis, and we asked them to take that sample before they treated the cows. And then, because we assumed from our pilot study that the majority of our mastitis that we were going to see in our big study was going to be caused by strep uberis, we asked them to start treatment with a green antibiotic, a penicillin, and in this case we used intracillin milking cow. So we asked them to treat with intracillin milking cow immediately after they took the sample. So in the case of a morning diagnosis, which most of our mastitis is diagnosed in the morning, so in the case of a morning diagnosed case, we asked them to take a milk sample, to treat with intracillin milking cow, to bring that milk sample straight into the clinic, to treat again that afternoon and treat again the next morning. So that cow received the standard dose of intracillin milking cow, 3 by 12 hourly treatments. That sample came into us, we plated it out immediately, and in most cases, in more than 95% of the cases, we had a result for the farmer the next morning. So that cow had received her third treatment of intracillin milking cow at morning milking on the second day. By 9 o'clock we had a result, and we could then phone the client and say, look, it was just strep uberis, or it was strep dysgalactia, or it was CNS. It was something that didn't require more treatment than your farmer had already given it. Or the other version was that it was Staph aureus. And we know that, or we thought we knew that longer treatments of stronger and longer drugs, and in this case we used pen clocks. We wanted to continue the course of penicillin and extend the therapy out, but we also wanted to add in cloxacillin just in case we had some penicillin resistance. So if it was Staph aureus, the cow had already received 1.5 days of intracillin milking cow, and then she went on to receive another 3 treatments at 24-hour intervals of the pen clocks, which meant she'd had a total treatment time of 5 days. One of the big things that we wanted to get out of the study, though, is we wanted to compare the overall results from a case-by-case prescribing method to our traditional days in milk prescribing method. In the days in milk, or our traditional herd-based approach, we just said if it was a cow that had calved in the last 7 days, well then we decided that that was most likely going to be strep uberis mastitis, and we'd treat with intracillin milking cow 3 by 12 hour treatments. If the cow had been calved more than 7 days, we were then starting to worry that maybe we were getting some more Staph aureus starting to come in at that time. So then we used a cloxacillin based product, and we used it 5 by 24 hour treatment intervals. So what we were comparing in the whole study was our case-by-case prescribing method of intracillin milking cow on every cow that got clinical mastitis, 3 by 12 hour treatments, and only adding in an orange antibiotic in the case of STAPH aureus, compared to our days in milk or our traditional treatment where we just assumed that cows that were recently calved were strep uterus, and we just assumed that cows that calved longer than that were STAPH aureus. Some of the results were really surprising to me. When I came over here from the Big Island, the first thing that I was told was that when you get mastitis in spring, it will be strep uterus, and we treat strep uterus with the shortest acting drug that we can, but it doesn't really matter which one it is because all drugs cure strep uterus pretty much the same. So we'd use either straight penicillin through intracellular milking cow or another really short acting product because we wanted that short milk withhold and getting the milk back into the vat as quickly as we could. And we weren't worried about cure rates because it didn't matter what drug we used. But the surprising thing to me that came out of the study was that the cows that were treated with straight penicillin, the intracellular milking cow, had a much better result and statistically significant better result than cows that were treated with the cloxacillin. What surprised me about that was that we had 3 times 12-hour treatment, so only 1.5 days of treatment with the penicillin, and we were comparing that to 5 days at 24-hour intervals of the cloxacillin. So we'd almost stacked the study against intracellular milking cow working in comparison to the cloxacillin product. But what we found was that actually the cows that received intracellular milking cow went far better if they had strep uterus mastitis than the cows that had had cloxacillin, even though that cloxacillin was for an extended period of time. In the case of STAPH aureus, and this was where we were really concerned about setting this study up, because we were told historically or anecdotally that there is penicillin resistance in our STAPH aureus. And so in our trial design, our study cows, our treatment cows, were getting 3 lots of 12 hours of intracellular milking cow, and if it was proven that it was STAPH aureus, they would get an extended therapy out to 5 days, but that 5 days was just 5 days of penicillin, only having the last 3 days with cloxacillin coming in from the pen clox. In the control group, those cows are actually getting 5 days of cloxacillin right from the start. So we thought the trial was potentially disadvantaging the product because it only had 3 days of cloxacillin. What we found when we actually went and analysed the results was probably the biggest surprise of all of the study, was that the cows that had received 5 days of penicillin plus 3 days concurrently, those last 3 days of cloxacillin, which was the intracellular milking cow followed by the pen clox, they cleared. And when we're talking about clearance rates here, we're talking about both a clinical and a bacteriological cure. So these cows had to pass a second and a third bacteriological test. That when we compared the results, the cows that had had the intracellular milking cow followed by the pen clox actually cured twice as well as the cows that had just had cloxacillin right from the start. And that was a big surprise for us. So the other thing that we found in the study was that overall, this concept of using case-by-case prescribing versus just using the herd-based system that we've traditionally used of cows being calved less than 7 days getting intracellular milking cow and cows calving later than that, getting something stronger and longer. The overall results from the study was that the cows that had been in the case-by-case prescribing actually did slightly better than the cows that were in the traditional approach. It wasn't statistically significant, but it was numerically slightly better. But the really big difference was that we used far less antibiotic. So in the case-by-case prescribing, not only were we putting the right antibiotic into the right cow for the right length of time and being responsible as we possibly could, but we used less total antibiotic overall. The other thing that was, and this was by trial design, but the other thing that we saw was that we used much less of the orange or red antibiotic in favour of green antibiotics. And with the move towards One Health and responsible use of antibiotics is a really important thing for us. It was one of the things we were trying to get out of the trial, and it was really reassuring that when we looked at the results, we'd actually managed to decrease the amount of antibiotic, but particularly decrease the amount of yellow or orange antibiotic that we'd been using in our clinic. We had great farmer engagement with this, and probably really important to start off with that. Our farmers became really engaged with the process of taking milk samples. And that led to a couple different things. It meant that because the farmers were taking milk samples and they had to do that in a relatively sterile way, we actually found that they treated their cows in a more sterile way. And I think we actually get better results when we treat cows in a sterile way. So it got the farmers to focus on mastitis. The other thing that it did, and this was from a veterinary point of view, it got the vets involved really focusing on the problem of mastitis and not just the solution. So it got us thinking about the whole farm situation at a much broader level, and it really brought in the engagement from our vets in what was happening on farm, rather than just a farmer coming in and saying, "I've got a case of mastitis, what should we use?" So while we've always liked to thought we've been engaged with our farmers, we saw a much higher level of engagement.
Milk sampling becomes a routine. It becomes part of farm protocol. We have 5 farm staff members, and once they all know the protocol of taking a sample before any drugs are given to cows, it becomes quite easy. You don't leave the shed without washing it. You don't insert a drug without taking a sample.
The farmers have saved money. We found our farms, and in this particular farm that we're on here now, this guy sends in every case of mastitis, and has done since we've done the pilot study. He sends in every case of mastitis because he says it's cost effective for him. What we charge to do the milk sampling and report back the results, he saves in, firstly, purchase price of the antibiotic. He's only buying 3 tubes instead of buying 5, but he's also got much shorter withhold and milk withhold period, being treatment and withhold after the treatment ceases. So he's actually saving money out of this. He's saving money, he's getting a better result, he's spending exactly the same amount with us, it's just in a slightly different area, rather than just buying on the drugs, he's paying actually for us to do the culturing and for some of the advice. It's meant we've become much more engaged with his problem, and he's become much more engaged with us, which has led to other advantages across other parts of our business, of course.
Well, we've done the cost analysis and it does come out at cost neutral for us, but it's a huge peace of mind knowing that you're giving the cows the best chance to be in the herd for the long term, and not just using a 2 strikes and the 3rd time you're out scenario. So you're looking at cow longevity, and you're looking at peace of mind knowing that you're putting the right drug into the right cow.
Based on our study, we only have intracillin milking cow and pen clocks on script for treatment of clinical mastitis. We try to use intracillin milking cow wherever practical. We like the idea of using a single active green antibiotic where it fits into the farmer's situation. But there are farmers where 24-hour treatment interval is really important. And there's other farmers who just can't quite get their head around leaving cloxacillin out just at the moment. So we do use pen clocks on script, but where possible, we keep intracillin milking cow as their primary mastitis treatment. So I suppose our take-home message from our study has been that we believe intracillin milking cow, or 1 gram of penicillin, is what we really need to treat clinical mastitis in New Zealand. We try and do that directly with intracillin milking cow where we can, or where the situation requires, we put in the pen clocks combined treatment.